Okay, this is a review for your membrane and membrane transport test. So first thing we're going to go over is membrane structure. So remember that the cell membrane is called the phospholipid bilayer. That's because you've got two layers of these phospholipids here. These phospholipids, remember, are very special because the heads, which are the orange circles, they like water. And the tails, which are these black lines, remember, they do not like water. So we'll talk about that more in just a second. The other thing you have a whole bunch of in your membrane is going to be proteins, and they can do many different things for us. So all of these blobs right here are proteins. You have some carbohydrate chains, which again we'll talk about in a second, and then there's a little bit of cholesterol in here. Okay, so when we look at, I'm sorry, I'm in the wrong spot. Here we go. Okay, so here are our phospholipids once again. So the heads are attracted to water. That means they're hydrophilic. And the tail down here is hydrophobic and is repelled by water. So since we have water inside of our cell and outside of our cell, we have our phospholipids lining up in this configuration with one row being right side up and the other being upside down. Okay. So again, here's our bilayer, and it's a gateway for things to move in and out of the cell. And it's the region here in the middle that's going to separate the two areas. So you can see molecules that are lipid soluble, like of course lipids themselves, can go straight through the bilayer. And then we have molecules like sugar and water and salt, molecules that are polar, meaning they have a positive and negative charge. They cannot get through the bilayer, so they're blocked. So we've got to come up with a way to let those molecules in. And one thing about the membrane is that it's really thin. So it looks thick and complicated, but it's relatively thin, and so that's going to allow some substances to get through, but not others. And so we call that being semi-permeable. So this is just another example as to how our polar molecules are going to move through. Anything that's polar is going to have to use one of these channels, and they're made of protein, and they're embedded in the membrane. One of the things about these channels is that they are specific to what they transport. So you can see that only one type of molecule is moving through. The channels you see over here to the right are aquaporins, and they transport water. When we look at the proteins that are in our membrane, these membrane proteins that go all the way through are called integral proteins or transmembrane proteins. And they have a polar region up here and a nonpolar region here. These proteins can do many different things. So we focus on these here, the channel proteins, but this is just showing us all the many other functions that these proteins can have in our cell. This one is of particular importance. This little orange chain that you see is a carbohydrate molecule. And if this carbohydrate is attached to a protein, we call it a glycoprotein. And if the carb is attached to the top of a lipid, we call it a glycolipid. And these are really important for cell identity. And I think I told you to think of them as cellular name tags. So, you know, when you go somewhere and you have to wear a name tag that identifies you, all the teachers in the school wear name tags so that, you know, that everybody knows that we belong here. And if a cell comes into your body without your name tags on it, then your immune system is going to go after that cell and attack it and get rid of it. Okay? So that presents a problem when people have to have organ transplants. And that's why they have to match up glycoproteins and glycolipids for the organ donors and, of course, the organ recipients. So there's some other proteins that are called peripheral proteins, and they're found on the outside of the membrane, and they don't go all the way through the membrane. So one is circled right here. Remember, your peripheral vision is your outside vision. It's the vision that's really important for driving. So remember I told you in class to look straight ahead, hold your arms out to the side, wiggle your fingers, and you can see your fingers even though you're looking straight ahead. And that's your peripheral vision. So I want you to think about peripheral proteins being those that are just on the outside, and many of them are going to be those glycoproteins. So when you see them on our diagrams, that's what they're going to be. And down here we have a transmembrane protein, which is going to be a channel or a pump, which remember we'll use for active transport. And then there's our aquaporin. Okay, this whole thing that you're looking at is called the fluid mosaic. That's just the official term, or just a way to describe the membrane. You always hear that with regard to the membrane. And we see all the different parts here. So our blue molecules here, those are our phospholipids. So this end likes water. Of course, this end doesn't. And we have a whole bunch of those arranged in 2 layers. These yellow things you see here, these are cholesterol molecules. So you've heard about cholesterol and people having high cholesterol, low cholesterol, whatnot. So we need some cholesterol. We just don't need too much of it in our body. But this helps make sure that the membrane is of the right fluidity, meaning that we don't want it too stiff and we don't want it too liquidy either. Over here, we have an integral protein, transmembrane protein, whatever you want to call it, and it's a channel protein because you see a hole here. This is just another integral protein. All these up here are peripheral proteins, and this one is a glycoprotein, and over here we have a glycolipid, which are some of those name tags that we were talking about. This is an aquaporin again, and the green stuff down here is the cytoskeleton. So here's our membrane. That's really it for membrane structure. That's all you got to remember. Top part's polar, the middle part is nonpolar. Anything that's polar has to go through one of these channels because it's blocked by this area. Nonpolar stuff like oxygen and carbon dioxide, alcohol, and anything that's lipid soluble, they can just zip right through here. But not polar stuff, it has to go through here. Okay, let's see. There we go. Everything's all labeled for us now. Okay. The membrane carbs, these were the glycoproteins, glycolipids. They are our name tags once again, and they are recognized by our immune system. And really, that's it for that part. So we're going to move on to transport. So here we are back to this slide where I started. And one thing about transport is that it's driven by the laws of thermodynamics. So you remember stuff is always moving. There's kinetic energy driving the movement of molecules in all environments. Remember, molecules in a gas move the fastest, and molecules in a liquid move next, and then molecules in a solid, they stay put. They really don't go anywhere. When we look at diffusion, we're looking at molecules moving from an ordered space to an unordered space. So this is our second law of thermodynamics. And in this picture, you can see our molecules of dye going from a high concentration over across to a low concentration. And we're going to do that until we hit equilibrium. And remember, that's the goal of any system. So anytime we talk about diffusion, we're going from high to low. Simple diffusion is high to low. It's a form of passive transport. And anytime we go from high to low, we do not need any energy, okay? And do not pay attention to this movement of water over here because osmosis is not a form of simple diffusion. So I'm sorry that is there, and I hope it doesn't confuse you, but just ignore that, please. Simple diffusion means that we're going from high to low without a protein channel, okay? Next is facilitated diffusion. This is the second type of diffusion. This is where we have to go through a protein channel. And we have to because these molecules right here are polar. And they can't just go through the membrane here. They have to go through a channel. They're still going to be going from high to low. And so no energy is needed. So there we go, high to low. And then the movement is going to be equal when we hit equilibrium. So when we hit equilibrium, remember, molecules will just move evenly back and forth. Okay. Oh, and don't forget, these channels, they do act like bouncers, and they will only let one particular type of molecule pass. So meaning sodium has its own channel, sugar has its own channel, potassium has its own channel. Okay? Active transport is the opposite. And now we're moving against the gradient. So we're going from low to high. That's like riding your bike from the bottom of the hill to the top of the hill, which you know means you have to pedal really, really hard to get to the top. You're going to have to use pumps to do this. So these pumps are going to move molecules from a low concentration to a high concentration. So low to high. And this is really low. I mean, it doesn't look like low to high in this picture, I know. But just assume there's a whole bunch more of these yellow things down here. So don't get confused by that. We're still going low to high, and we got to use ATP. And these little pumps, you see how they kind of look like rocking chairs? That's how they're structured. So they look a little different than our channels. Active transport is going to move all kinds of things. We talked about the sodium-potassium pump, which the sodium-potassium pump moves 3 sodiums out and 2 potassiums in. So it is a pump, just like this little thing, and it's going to be moving molecules from one side of the membrane to the other against the gradient, going from low to high. And then last, for active transport, we have endocytosis and exocytosis. This is just showing endocytosis. So endo means going in. This is bulk transport, you know, like you buy stuff in bulk at Sam's. So we're moving big molecules. And, you know, when you move big stuff, it takes more energy. So that's what we've got here. We've got 2 types. Phagocytosis is for food particles that are larger, and pinocytosis is for droplets of fluid, something that's a little bit smaller. So, and then you don't have to know this down here, so just ignore that picture. Okay? So, just to sum it up, what we've said so far is that you can move across the membrane in 2 ways. You can do active transport, or you can do passive transport. Passive transport, you're going from high to low with the gradient, no energy. And that's either simple diffusion, facilitated diffusion, or osmosis. So we'll get to that in a second. Active transport, you're going against the gradient. We're going from low to high, and so we have to use a pump. We know that this also includes endocytosis and exocytosis. So here we go again. Simple diffusion, because there's not a protein channel, high to low. Facilitated diffusion, there is a protein channel, still high to low. But remember, facilitated is transporting polar molecules, molecules that have a charge. Down here at the bottom, we have active transport. We're using a protein. We're going from low to high, so we have to have the energy. Okay. Now we're going to move to the last thing about bulk transport. This is exocytosis. It requires energy too. So that's what you see in the picture down here. We've already talked about endocytosis. Whoops. So this is going to cost us energy as well. Okay, so don't forget about endo and exo. And now for water. So this is all about osmosis. And I want to just remind you that when we deal with osmosis, we're dealing with water. And, you know, there's all kinds of stuff dissolved in water. So no matter, you know, what you like to drink, whether it be, you know, like me, Diet Coke, coffee, tea, Gatorade, whatever, it's a water solution. So solutions are made of solutes and solvents. So the solvent is water, like right here, and the solute would be this powder, which would be dissolved in the water. Okay, so just remember the difference in your solutes and solvents. Solutions are of different concentrations, and I talked about coffee the other day, and I said, you know, I told you that regular Starbucks coffee is too strong for me, and so this is an espresso machine. And, you know, espresso is served in really, really tiny cups because it's so strong and you just can't drink that much of it at one time. So, of course, you know, they're going to make your, you know, fancy lattes and things like that with this, but they're going to mix some other stuff in it. But right now, if you were to drink this, it would be really, really strong to you. So think of that like hypertonic. Over here, this is weak coffee. And see, it doesn't even look like coffee. It looks more like tea. So it's much lighter in color. It doesn't have as much solute. So think of this like hypotonic. Hyper means more. Hypo means less. Okay? So kind of keep that in mind as we move through this. And so osmosis is still diffusion. It's just diffusion of water. It's moving from high water to low water, which really means that, that we're going to be moving water into our hypertonic solution because we're trying to even it all out. So that is the osmosis rule, people. Water moves into the hypertonic. And if you remember that, you can answer these things. Okay? So here we go. Hypertonic is more, like our strong coffee. Hypotonic is less, like our weak coffee. Isotonic is the same. So here we have hypotonic here. We don't have as much solute. We have hypertonic here. We have a lot of solute. So we're going to be moving our water from this side to this side, okay, into hypertonic. And that affects plant and animal cells differently. Remember, animal cells, if you put them in hypotonic, they're going to burst because they don't have a cell wall. Plant cells won't. So plant cells like to be turgid. So they're not going to blow up in a hypotonic environment. Isotonic environment is what our cells want. Plant cells are they're okay with isotonic. It's not their favorite, but they're not dead either. So they're just kind of okay. And hypertonic solution causes both to shrink. So down here, remember this is called plasmolysis. So think about these examples and remember there's a difference for the plant and animal cell because of the cell wall. So in a hypotonic environment, water is always going to go in and cells are going to swell. And if you are a little protist, a little protozoan that lives in the water and you only have one cell, you could potentially blow up if you take in too much water. So if you're in hypotonic, you could blow up. And so that's why we have specialized structures like in this paramecium that's going to pump extra water out. Okay? So hypotonic makes cells swell. Okay. Hypertonic makes things shrink. So it makes them shrivel up. So salt makes things shrivel up. And so we've got the animal cell here. We've got the plant cell here. That is shriveling in the hypertonic environment. And then we have isotonic, which for animal cells is perfect. And plant cells, it's okay. These are the men that discovered aquaporins, which are our water channels. And so remember, water goes from high to low. It's going to be going through these little structures. Remember, just like we said, water is going into hypertonic. Okay, so take a second, look at what we've got. You saw this in class. We have a cell in an environment. We're going to pick out if the cell is hyper or hypo, if the beaker is hyper or hypo, and then which way does the water go. Okay, so for the cell, we have a 0.05 molar solution because you just add these two things up because it's still sugar, so it's still a 0.05. Over here we have a 0.03, so that means that my cell is hypertonic. So if the cell is hypertonic, the beaker is hypotonic. And remember, water goes into hyper, so water is going to go in. Okay? Guys, that is just about it. Just make sure you read over your notes. Look at your flashcards again. Look over your notes. I'm going to post you a Kahoot and probably a Quizzes to play as well. So just do your best. And if you have any questions, just send me an email or whatnot, or just talk to me before class. Okay? All right. There you go.